Hello everyone and welcome to week 11. Lesson 11 is about ELISA testing. This week's lab is about the diagnosis of infectious disease. So there's a few categories of the diagnostic methods. The first is the growth-based biochemical tests, like what we've been doing in the lab so far to this point. So these are the tests that detect the bacteria from the chemical reactions, the products of its chemical reactions. So, for example, the gas production or acid production or enzyme catalyzed reactions as a result of the bacteria that we use to diagnose and identify it. So, for example, the selective and differential media that we've used so far in the lab to diagnose bacterial infections. So for example, the water testing lab, the staph and strep identification lab, the enteropleurae lab, we've used growth-based biochemical tests for identification in all those examples. The next few tests you might be familiar with and recognize because of COVID. So let's go through those. The first is the serological tests. Now, serological tests focus on antibodies in the blood. So, for example, this is a blood test that can be done to detect the antibodies to COVID. And that's done to determine if someone has had the infection in the past. So maybe that they didn't know it if they were asymptomatic, or maybe if they were not tested at the time. Serological tests like the ELISA test for COVID-19 antibodies can determine if a person has antibodies for the infection, indicating that they had this infection in the past. So that's the blood test that you've heard of for COVID-19 antibodies. The next one is the nucleic acid tests. And this one is the test, the nasal swab test that's done to diagnose a current COVID infection, for example. So the PCR test for COVID-19 diagnosis is detecting that viral RNA in the person's sample taken from a nasal swab. So that's a very accurate test to detect a current COVID infection. And that's a nucleic acid test because it's looking for RNA of the pathogen. Then the next one is the antigen test. The antigen test, for example, is the rapid COVID-19 test. So that works by detecting pieces of the proteins that are found on the pathogen and looking for those pieces of the protein on the pathogen surface to determine if the person has an active infection with that pathogen present. So that's a faster test. It can be done in a matter of minutes. It's not quite as accurate as the PCR test. The PCR test is kind of the gold standard for COVID-19 diagnosis, but the antigen test is the rapid one. So I wanted to go through all of those so you're familiar with them in terms of what's going on now with COVID and the different types of tests that are available. So those are all the categories of diagnostic methods for diagnosing an infectious disease. But this week's lab, we're going to specifically look at a serological test, and we're going to perform in the lab a simulated serological test, an ELISA test, to detect antibodies. So this week's lab is going to focus on the category of serological tests. So what are serological tests? They are blood tests that look for antibodies in the blood. So our body has 3 lines of defense against pathogens. The first is the physical barriers, like our skin, mucus, even earwax. Those are all physical barriers to pathogens to prevent them from getting inside our body. So that's the first line of defense. The second is the nonspecific immune response. So that's the body's nonspecific reaction to a pathogen. Like getting a fever, for example, is a nonspecific immune response to a pathogen to try to kill that pathogen with a fever. So that's an example of a nonspecific immune response. And then the third is the specific immune response, and that is specific to the type of invading pathogen. So to understand more about the specific immune response, there are two important terms to know: antigens and antibodies. So antigens are a substance that is foreign to the body that produces an immune response. So for example, a protein on the surface of a pathogen is an antigen, and it causes an immune response in your body. It's an unknown or foreign substance to your body, produces a response. The antibodies are blood proteins that are produced in response to a specific antigen, and they're capable of binding to that specific antigen in order to help destroy it and fight an infection. So antibodies are these Y-shaped proteins that you can see in the picture here, and they have specific antigen binding sites at the end of the Y shape, and that's where they bind to the particular antigen that they target. So this topic is really relevant to our everyday lives right now because we hear all the time about COVID-19 and COVID antibodies and how having the antibodies can potentially help to protect a person from being reinfected, or that how antibodies can even be used as a treatment for COVID-19. So antibodies are made by your body in response to the specific antigen. So for COVID-19, that antigen is a protein on the surface of the coronavirus. And your body produces antibodies in response to that specifically targets that antigen. And those antibodies then circulate in your blood to help destroy that pathogen if you're exposed to it again in the future. So antibodies are specific to their antigen. They bind only specifically to that antigen that they were produced to target. And because of that, they can be used for diagnosis of that particular pathogen. So ELISA testing, or enzyme-linked immunosorbent assay, is a test used to detect and measure antibodies in the blood. Now we're going to go through the steps to doing an ELISA test. So I've been talking a lot about the coronavirus, but ELISA testing can be done for all different kinds of infectious diseases to diagnose them. So in the lab this week, we're going to be doing an ELISA test. It's a simulated ELISA test so that we're not actually working with any dangerous infectious diseases, but it's a simulated ELISA test for the diagnosis of HIV. So in the first step of this test, a known antigen is added to the wells of this 96 well plate, and then it's incubated so that the antigen can bind to the bottom of the wells, as you can see in the picture at the bottom here. So in this experiment, for the diagnosis of HIV, the antigen is an HIV protein. So that gets bound to the bottom of the wells. Then excess or unbound protein is washed away with a detergent. And the detergent also serves as a blocking agent, which means that it holds as a thin film on all the remaining binding surfaces of the plate so that it prevents non-specific binding. In the second step, the patient's blood serum is added to the well and incubated to allow the primary antibodies, the person's antibodies that are in their blood serum, to bind to the antigen. And then the excess or unbound antibodies are washed away. So the primary antibodies are specific and only bind to the specific matching antigen. So what's actually happening here? If the patient is infected with HIV, that means their body has started producing those specific antibodies to target HIV. So that means that their serum would have those primary antibodies in it that will bind to the HIV antigen in the well. And it would form these antigen antibody complexes, as you can see in the picture below. But if the person is not infected with HIV, there are no antibodies that have been produced, so no reaction would be occurring. In the third step, enzyme-labeled secondary antibodies are added to the wells and incubated, and they bind to the primary antibodies. And then again, excess or unbound material is washed away. So these secondary antibodies are general, and they're not specific. Like the primary antibodies are specific and only will bind to that particular antigen that they target. But these secondary antibodies are general, and they will bind to any primary antibodies. So you can see in the picture below, the secondary antibodies are the white Y-shaped antibodies with that star on top, and that star is representing what they are labeled with, which is Horseradish Peroxidase enzyme, and that is going to make detection of these antibodies possible. So what's happening in this step is that it's binding with a labeled antibody so that we're going to be able to detect the presence of the patient's antibodies. So I told you in that third step that the secondary antibody has this enzyme attached to it that's represented by that star, the horseradish peroxidase enzyme. And this fourth step here gives that enzyme something to do. So the fourth step is a chromogenic or color-producing substrate is added to the wells, and it's incubated to allow color to develop. So in the presence of hydrogen peroxide, which is included in this substrate, that horseradish peroxidase enzyme catalyzes the oxidation of the chromogenic substrate. And that reaction forms a colored product. But remember, that's happening only if the patient is positive and has those primary antibodies. So if the patient has the primary antibodies, they bind to the antigen, and then those secondary antibodies bind to the primary antibodies. They have the enzyme attached, and when this chromogenic substrate is added, that enzyme can change the color from clear to purple. So that's how we can actually detect that the antibodies are present. So some wells you might see are darker than others, and I'm going to pose that to you as a question in the post lab of why you think that might be. So I just want to summarize with this figure what's happening in an ELISA test for a positive patient. So in the first step, we have the antigen that gets bound to the bottom of the wells. And in this case, the antigen is an HIV protein because we're performing an HIV test. So in the second step, the patient's serum gets added to the wells. And if that patient is positive for HIV, they're going to have the primary antibodies that will bind to the HIV antigen. Now, we don't have a way of seeing that with the eye, so the ELISA test uses the secondary antibody, which is general and binds to a primary antibody. The secondary antibody is labeled with an enzyme, so that in the fourth step, we can add a chromogenic substrate that will change from colorless to purple when it's oxidized by that enzyme. So that allows us to see it with the eye, whether the patient is positive and has those antibodies or whether they're negative. So I just wanted to also include here that rapid test strips are also available for the detection of HIV antibodies. And I also wanted to mention the importance of positive and negative controls in these tests, because you would want to check with positive and negative controls to make sure that your test is working correctly, your reagents are working correctly, because we absolutely would not want to give false results to a patient.